investigates neural mechanism of daytime sleepiness. So, what is sleepiness? In a simple term, it's a biological drive to sleep. But it's actually a very complex phenomenon, which is an output of interaction of sleep-promoting and wakefulness-promoting systems of the brain. So there are many causes of sleepiness, with most common one being sleep deprivation that may seem natural and adaptive, but it can also be a pathophysiology of one of the sleep-wake regulating circuits. So what are these circuits? Besides cortex and thalamus that are obviously involved, there's basal forebrain and a set of nuclei in the hypothalamus and brainstem that we call ascending arousal network. And they play a key fundamental role in neuromodulation. So animal work has provided us extensive knowledge on anatomy, function, and projections of these nuclei. However, animal work is limited in studying them simultaneously at a circuit level, whereas, on the other hand, the human brain imaging is limited by their small size and deep location in the brain. So in this project, we employ a state-of-art high-resolution imaging at ultra-high field to address that challenge and investigate sleep-wake regulation circuits and underlying daytime sleepiness. So we asked 27 participants to restrict their sleep to 4 hours prior to the MRI session, and ask to conduct for about 1 hour a very simple attention task while we collect high-resolution images of their brain. And using newly developed segmentation tools, we were able to identify these nuclei of the ascending arousal network in the individual anatomical space, then extract time courses of each of these nuclei and relate it to the behavior. So luckily for us, subjects were very drowsy and committed around 15% of these so-called omission errors, where they failed to respond to stimulus. So that gave us opportunity to lock brain activity to either first omission trial, that represents loss of responsiveness, potential microsleep onset, or first correct trial after an omission trial, that represents this regaining of responsiveness, arousal from a microsleep. So with respect to the loss of responsiveness, we investigated signal from cortex, thalamus, basal forebrain, 3 nuclei in the hypothalamus, and 8 nuclei in the brainstem, including locus coeruleus, and found that all brain regions show consistent decrease of bold signal during loss of responsiveness, except for one, the hypothalamic nuclei in the preoptic area that is actually known for being the only sleep-promoting brain region in the brain. With respect to regaining of responsiveness, we found the opposite. All brain regions show increased bold activity in line with their wakefulness-promoting role, and again, except for the preoptic area that showed decreased activity. So next we wanted to test if these brain activity vary in time. So we extracted the timing of each significant peak and trough, and our results revealed that the timing of this activity is similar between the two conditions, but they're actually distinct between the nuclei, with hypothalamus leading the changes, brainstem following, thalamus and basal forebrain being the last. So taking together, our results suggest that brain activity from ascending arousal network can be reliably detected with fMRI, and their diverse pattern of activity suggests that this may be a local neuromodulatory effect on the fMRI signal. If you like to see more results, have any questions or comments, please visit my poster A54 today at 3 p.m. Thank you for your attention.